Hey gang, welcome back. We are going to start learning about the structure of DNA, which is really important because it's the structure of a molecule that gives it its function. So the structure of DNA is the key to understanding how it works or how our genes work. And recall that DNA is our genetic material, and there's a few things genetic material needs to be able to do, right, to be able to pass on information about our genes. So one thing is that it needs to be kind of a tough material or rugged. It shouldn't be able to break down easily. And it also needs to be complex enough to carry a code. Right, a long time ago when they were trying to figure out what our genetic material was, they knew it couldn't be a sugar or a fat. Those were too simple. It needed to be able to pass on information by having some sort of internal code, kind of like, how Morse code has dots and dashes, or computer code uses zeros and ones in a different order to mean different things. So our DNA needs to be able to do that as well. So DNA is made out of nucleotides, and this is the basic structure of a nucleotide. So it always has a phosphate group, and this is the same sort of phosphate group that we saw on an ATP molecule. So nothing new there. It's just a phosphorus with some oxygens around it. And then in the middle is a sugar, not just any sugar, it's a very specific sugar. It's called deoxyribose. Ribose is a 5-carbon sugar. The carbons aren't all shown here, but there's one here. So this is number 1, number 2, number 3, number 4, and here's the 5th carbon up here. And it's called deoxyribose because normal ribose has an extra oxygen group hanging off here. This one doesn't. So deoxyribose is just the specific way of naming this particular sugar. And you might recognize that that's part of the name of DNA, deoxyribonucleic acid. So here's where the deoxyribo part comes from. And then here is a nitrogenous base, meaning that it has a lot of nitrogen in it. And there are 4 of these. So any 4 of these could possibly be connected to the sugar right here. And these 4 bases are called cytosine, thymine, guanine, and adenine, but we normally just abbreviate them as C, T, G, and A. So any one of these bases connected to a deoxyribose, which is connected to a phosphate group, makes up a nucleotide. So in a second, I want you to pause the video and go grab a handful of these little ball and stick model thingies. And you are going to make the 4 bases. So the white here is the sugar, the ribose. And you're going to attach on a phosphate, which is the red one. So it's going to go down into the sugar like that. And then you're going to add one of the bases. They're different colors. So yellow is A, blue I think was C, orange is T, and G is green. So you're going to take that base and plug it into the sugar. So it's not going to make a nice line, but it's going to make more of a triangle. It's a bent shape. And make sure the whole of the base is on the outside. So you're going to plug that into the sugar molecule on the side. So it's going to look just like this, with phosphate on top, sugar in the middle, and the base on the outside. And you'll want the holes available on the outside of these guys. So they're going to plug into the white in the middle. So go ahead and pause and make 4 of those, one for each of the bases. Okay, so hopefully you're back now, and we're going to move on to how these get put together. So DNA is a polymer of nucleotides. So remember, a polymer is a large molecule made up of lots of repeated smaller molecules. So DNA is just a polymer of these nucleotides all hooked together with covalent bonds. So we're going to model doing that as well. And the way this happens, here's one nucleotide, and then here's another one coming up to join it. So there's an OH group on this phosphate, and it's going to come and hang out up here next to the ribose. And these two hydrogens and this oxygen are going to run off together. And you know what they make? H2O, right, water. So they're going to go off and become water, and this oxygen here is going to covalently bond with the phosphate. So you're going to end up with a nice backbone of phosphate, sugar, phosphate, sugar, phosphate, sugar, with the bases off to the side. So it'll look something like this. So go ahead and do that. Grab your nucleotides. And so your red phosphate on one nucleotide is going to come join up with the sugar on the other. So hopefully you can see this here. Here's my one nucleotide, and that phosphate's just going to come up and pop together. Hey, you polymerized. Good job. Okay. And we're going to bring up another one. So get all four of yours hooked together. Where's my phosphate here? Okay, got to get my phosphate and sugar. Snap. And last one, phosphate with sugar. Snap. Let's go ahead and pause if you need to and get your DNA strand made. Yay! All right. So another piece of the puzzle of what the structure of DNA was figured out by this guy named Chargaff. And he purified DNA from lots of different organisms, and he was studying the bases. So he looked at, in the DNA strands, in the polymerized molecules, he found the percent of nucleotides with an A base, the percent with a T, the percent that had a G base, and then the percent with a C. And then all of these percents should theoretically add up to 100, right? So 100% of the DNA. And he was looking at all these numbers, and he found a very clear pattern. So I want you to take a look at these numbers and see if you can find it. So they come in pairs. There's always two that have a very similar percent, and then the other two also have a very similar percent. Do you see it? So this is close to 30 and this is close to 30. And then these two are close to 19. So it's not a perfect match, but in all of these, if you look closely, these are very similar to each other, and these are very similar to each other. These are 27.6 and a half average, whereas these are both really close to 22.4, right? So we saw this in every organism, and when you put them together, like the percentage of A, B, and T together wasn't always the same in every organism. That didn't matter so much. But A and T were always similar, and G and C were always very similar, very, very close. So he came up with this rule. Chargaff's rule is that in DNA, the number of adenine bases equals the number of thymine bases, and the number of guanine bases equals the number of cytosine bases. He wasn't sure exactly what that meant, but he knew that it meant they must be paired up somehow. So for every A, you're going to find a T, and for every G, you're going to find a C. So it took a couple more pieces to figure out why this was. So one of those puzzle pieces was an X-ray picture made by Rosalind Franklin. So she also isolated DNA, stretched it out, and took X-rays of it. And then this was her most famous one. She saw this, and she said, aha, that's the exact same pattern you would see from a spiral or a helix shape. So she saw this X-ray pattern, and she knew that DNA had to have a helix shape. And then there were two other guys working on trying to figure out the structure of DNA at the same time, and they put lots of different models together, and none of them worked right until they saw this X-ray picture, and then they knew, aha! DNA is a double helix, meaning that there's two strands of DNA that swirl around each other, so they make this nice double spiral thing. So it's called a double helix. And this part here is the sugar phosphate backbone. And then in the middle between the two strands are the nitrogenous bases. And so this explained Chargaff's rule as well. So on one strand where there was an A, it would interact with a T on the other strand. Wherever there was a G on this strand, it would match up with a C on the other strand. So it made this really nice structure with one strand running one direction and the other one kind of flipped over running the other direction with the bases all paired up in the middle. So this is called anti-parallel strands, meaning they're running in opposite directions. And it follows the base pair rule of each A matching with a T and each C matching with a G. Now, one thing that took them a little longer to figure out was that these matching bases were held together with hydrogen bonds. So hopefully you remember these from learning about water. A hydrogen bond is a weak bond. You know, it's not nearly as strong as a covalent bond or an ionic bond. But it's sort of a weak attraction between an oxygen on one molecule and then a hydrogen on a nearby molecule. So guanine and cytosine, when they are paired up next to each other, they can actually make 3 hydrogen bonds between them. And then adenine and thymine also make hydrogen bonds between them. So just the way that these bases are set up physically, they just match with each other perfectly. So A always matches with a T, and a G always matches with a C. And if you try to put like a G with a T, they're not going to form those hydrogen bonds. So this is really cool that hydrogen bonds are what hold the two strands of DNA together. We're going to find this out in the next module. It helps with how they are able to make copies of themselves. All right, so at this point, this is where we're at the end of this video. Hopefully you have your little single strand DNA. I want you to find a partner that also has a strand of DNA, and you guys are going to put them together. So find the other strand, and these little clear rods, I don't know if you can see that, that's the hydrogen bond. So you might have to take one strand apart so that you can do the base pairing right. But see, if this is a G on one strand, then I've got to find what on the other strand? A C, right? So C's are blue. So I would take this apart. And make sure they're going in opposite directions. So I've got sugar and then phosphate on this side. So I'm going to do phosphate and then sugar on this side. And then my C is going to match up and make a hydrogen bond with my G. So go ahead and close the video and work with your partner to make a double strand of DNA.